Okay, so students, today we are going to talk about the important property of amino acids called the isomeric properties or optical chirality and optical rotation and stuff like that. So you can read, if you want to, if you can read more about this in Leringer's book, the one that was referred to you, page 15 to 20, for any further reading. So what you can read more about what is configuration and conformation. So we'll discuss that, um, and so on. Okay. So let's just understand what we are going to discuss today. So we are going to discuss something called isomerization and what isomerization is about. Okay. So what are the various types of isomerization one can encounter in a molecule? So first, one kind of isomerization that would be you may have encountered in your plus 2 is the structural isomerization. That is one type, and that is structural isomerization. And the second kind of isomerization you can, which is to be more relevant to what our lecture is the stereo isomerization. So let's just discuss structural isomerization first. So structural isomerization is something that simply happens due to change in the structure. Okay. So what is an example of this? So this can be three types: chain, position, and functional. So chain means the backbone carbon chain. If something changes in the backbone carbon chain, it's called the carbon skeleton, that leads to structural isomerization. For example, this methyl group here, this CH3 group, that moves to this position, and this hydrogen that moves to this position. So this methyl group is right here, and this hydrogen has moved to this position. So this is a structural isomerization, butane versus 2-methylpropane. Positional isomerization is some functional group, like for example double bond here, they change the relative position in the molecule. So here if you look at the numbering, the double bond is between 2 and 3 carbon. In this molecule the double bond is between 1 and 2 carbon. Okay. The third type of isomerization is functional isomerization, where a particular functional group is completely changed, okay, without changing the chemical formula. So isomerization, they all isomers have same chemical formula, but you can see this particular one is a double bond here, but this one has a cyclobutane. So the functional group is completely changed, but the chemical formula remains unchanged. Okay, so that is functional isomerization. So in amino acid side chains, we can see one example we can show here, and you can tell me what this is about. So we have isoleucine and leucine. So the main structural backbone, that is the alpha amino group, which is right here, and the alpha carboxyl group, which is right here, their positions are the same, alpha amino and the alpha carboxyl. But you can see there is change in this part. The chemical formula remains the same, but there is change in this part. So what kind of isomerization is this? Okay, so we will discuss that after this slide. The other type of isomerization is the stereoisomerization. So stereoisomerization deals with change of the functional groups or atoms in the 3D space. Okay, so one classic one is the cis-trans isomerization, and second one is the optical isomerization. So we'll discuss both of this in the next slide in more details. Okay, so coming back to this particular problem, so this particular isomerization is structural isomerization, and more specifically chain isomerization. Okay, because you can see the carbon backbone is rearranged. So the CH3 group is moved to this position, okay, and the hydrogen from here is moved to this position. So this is a chain slash structural isomerization. So let's discuss the stereoisomerization, which is of our interest in more detail. So stereoisomerization, as I mentioned, is among molecules that have identical chemical formula, but the atoms in 3D space are differently connected. Okay, so this particular one is an example of an amino acid. More specifically, you should know from memorization your structures. This is alanine. So in alanine, this is the relative arrangement of groups in the space. So this is the alpha carbon, this is the alpha amino, this is alpha COOH, and if you change the NH2 groups with the hydrogen, that would lead to a different stereoisomer. So I'm going to draw it. So we'll soon learn that this type of bond is called wedge bond, which means this is facing towards you. So I'm changing the relative positions of the atoms in terms of the wedge bond, while keeping the acid group and the side chain same. The dash bond is the ones that is away from you. So they are being kept the same. So I have changed the NH2 and H. So these two are stereoisomers. So what kind of stereoisomers, that we'll learn in a moment. So that is, but these are structural isomers. Okay. So we will learn a term called configuration in this regard. So configuration precisely means that the change of 3D arrangement of atoms in space, but by breaking a bond. Okay, so it's a relative 3D arrangement of atoms in space which cannot be changed without breaking a covalent bond. Okay, so you have to, for this to happen, for this interchange to happen, you have to break the NH2 bond and move it to the hydrogen's place, and vice versa. This is opposed to conformation, where you can change the 3D arrangement without breaking a bond. Okay, so that is the major difference. So first, let's discuss the geometric stereoisomerization, or more commonly called cis-trans isomerization. So cis-trans isomerization is something you may know from your +2. So if you have a double bond, for example, and the groups are oppositely placed with respect to the double bond, this is called trans. If they are placed on the same side of the double bond, that is called cis. So just want to generalize this a little bit. So this just not just applies to a double bond, but this also applies to any restricted bond rotation. So for example, in a ring, so ring refers to something called a restricted bond rotation, because this is a sigma bond, but you cannot rotate this at your will. So this is not possible, right? So if this was without a ring bond, like a simple sigma bond, then you could have rotated it. So I will show an example. First, let me erase the bond rotation. So this particular bond is towards you, and dash bond is away from you. Similarly, in this molecule, the CH3 is towards you and H is away from you. In this molecule, this chemical formula will be the same, but I am interchanging the relative arrangement of the groups. So H in this carbon will be towards you and CH3 away from you. Now if, because this bond rotation is not possible, this is a restricted movement bond, a restricted rotation bond. So now the CH3 atoms, CH3 groups in this molecule are in a cis arrangement, whereas CH3 groups in this molecule are in trans arrangement. Okay, so they are, because they are oppositely placed with respect to this particular bond. So why I am referring to the fact of restricted rotation, because think about this same molecule in a non-cyclic molecule. This is CH3, this is H, this is CH3, this is H, and this is some other carbon here, okay, with some other functionalities. This is some other carbon here, some other functionalities. Okay, the same group on this side, CH3, H, and CH3. Okay, other carbons here and some other carbons here. So this can be interchanged by simply rotating this bond. Okay, so there is no restriction placed here. You can rotate this and you can do this. So this are not geometric isomers, but these two are geometric isomers. Okay, so this is geometric isomers and these are not geometric isomers. Okay, I hope that is okay. So this you can place as conformers because the relative arrangement can be changed by 3D arrangement, by bond rotation. Okay, so these are conformers. Okay, but these are not cis-trans isomers. These are so you can say these are not configuration. Okay, so these are cis and trans configuration, geometric configuration, or isomers. But these are not geometric isomers or configurations. So I hope I made the differences clear enough. Okay. So one practical example of this is in terms of something that happens in our eyes. That is this particular molecule called 11-cis-retinal. So 11-cis-retinal refers to this 11-12 bond, where this bond is cis with respect to the 11-12 double bond. Once you put light in it, and light contains photon, this leads to temporary breaking of the bond and then bond rotation. Okay, so this becomes temporarily single bond. So this is the 11-12 position, and it rotates. Okay, so it becomes a radical and it rotates to form a trans. Okay, and the radicals then form the double bond again. Okay, now it becomes a trans retinal. Okay, so all the other bonds here, so you can tell, so this particular one is a trans, this double bond is a trans, this double bond is a trans, but that particular one used to be cis, now it is a trans. So change from cis to trans sends a signal to our eye via a nerve impulse that a light has been absorbed, and this leads to visualization. Okay, so this is part of the process of visualization. But so this is an example of how cis-trans isomerization is important in our eyes. Okay, so one more example is the EZ isomerization, which is similar to cis and trans. I'll quickly go to one slide and I'll discuss this. Okay, so EZ isomerization. So EZ isomerization is CH3 and CH3 group, and let's do this on the other way, CH3 and CH3 groups. Okay, so if you look at the letter E, so if you send a line across this, okay, so this travels from this direction, this travels from this direction, this travels out in the opposite direction, meaning the arrow's direction gets changed. So this particular one is called E. Z, if you draw, so this Z, if you draw like this, Z goes like this, and goes out in the same direction. So that refers to this molecule that the direction remains changed. So this is a Z. Okay, so Z means same. So this particular isomer is called E, and this is called Z. So this is another way of referring to molecules in addition to cis and trans. So EZ isomerization. Excuse me. So so that represents the cis-trans isomerization. So now we are going to discuss the optical isomerization. So before we learn the optical isomerization, let's learn something about Fischer projection and wedge dash drawing of bonds. Okay. So I already discussed wedge means a bond that which is drawn in a bold line, and it reflects to a bond that points towards you. Dash is a bond that is pointing away from you, and this line simply means bond that is on the plane of the paper. So this is basically a universal way of representing the bond. So first learn what is a Fischer projection. So Fischer projection is invented by scientist Fischer, who is one of the pioneers of stereochemistry. Okay, and what it refers to is this particular type of arrangement is called a Fischer projection. Okay, or a particular way of drawing. So if you have A, B, C, D present around a carbon, this traditionally, this particular arrangement is, or way of drawing, is called the Fischer projection. And in terms of 3D representation, what it refers to that the vertical bonds, that is A and C, are dash bonds, that is away from you, and this bonds D and B are towards you. Okay, so that is a Fischer projection. Okay, so this is again a universal, as a universal way of representation. So anytime you have this arrangement, this always refers to the vertical ones away from you and the horizontal one towards you. Okay, so that is a Fischer projection. So let's just next move to something called chirality and see what is the implication of Fischer projection representing is. So chirality is, so this particular molecule is drawn in a Fischer projection way. Okay, so when all four groups of the carbon's valency are non-identical, this molecule is, or this carbon is called a chiral carbon. Okay, and it is represented with the star. One more time, when all four substitution of the carbon's four valency are non-identical, it is refers to a chiral carbon, and this is represented with a star. Okay, so what is so special about chirality? So we should understand this from the perspective of two aspects: A, how it interacts with the light, and B, how it interacts with other biomolecules. Okay, so again, this particular molecule is drawn like this because this is how the 3D arrangement of this molecule in Fisher projection should be. So this is Fisher projection, and this is 3D arrangement in the wedge dash model. So first thing we want to know how it interacts with the light. Okay, so so for that to understand, we have to consider a special kind of light called plane polarized light. So what is a plane polarized light? So when you have a normal light source, this light has the light waves or electromagnetic waves oscillate in all possible directions, meaning that this light, I'm just redrawing it. So this is the light source, okay? So, and this is x, y, z plane, x, y, and z plane, okay? As if your eye is, think about this is here. So what happens here, this light can, one wave can oscillate in the x, y plane, okay? So your eye, because it is placed in this way, your eye is visualizing this in the xy plane, okay? So let's draw this out. This is x, y is this axis which is towards you, so you won't be seeing it, and z is placed perpendicular to it, okay? Z and minus z, and x and minus x, okay? So as I said, it's in all direction, meaning it will also have another light which is, let's say, the yz plane. So let's draw this that way, yz plane. So you are going to see this like this with the yz plane. So that oscillates in the yz plane. Okay, so it is rotating like this, or oscillating like this. Okay, xy is oscillating like this, and yz is like this. Okay, so let's just do one more form of light, which would be, let's say, in angular plane, which is neither xy or yz but in between. So let's just do that like this. Okay, so in this circular picture it would be like this. Okay, so another one, let's just say with the yellow color we can draw it like this. So that is how light oscillates in all possible directions. So one of them like this, one of them like this, one of them like this, and another one like this. So this is a normal light. When you pass it through a crystal called polarizer, the light then, depending on the axis of the crystal, it lets only one plane of the light go through, and let's just, for our example's sake, call the vertical one it lets pass through, but not the other one. So now only the vertical one in the xy plane is being let through, but the all others are not allowed. So this is now called a plane polarized light because it is oscillating in one direction. Okay, so this is the plane polarized light. So what is it between the plane polarized light and an optically active molecule or a chiral molecule? So chiral molecule, when it interacts with a plane polarized light, what it does, so this, when it interacts with a chiral molecule, it leads to rotation of the plane polarized light. Okay, so this, let's say the initial plane was like this, so now it will rotate to a certain degree and it will cause a angle change. Okay, so let's just call this angle is phi. Okay, so that is the one rotation. No, sorry, the rotation of the plane polarized light. So this is a property of chiral molecules. So that is why they are called optically active molecule. Okay, so what is the special part of the optical molecules and biochemistry? So you are going to find chiral center in almost every molecules in bio. Okay, so that is really important. Okay, so we'll see many examples, and we'll see that optically active molecules are ubiquitous. Okay, so that is how, that is one aspect. So how what is the interaction between a plane polarized light. Second thing is that um, so the other point that I want to mention, that this is basically measured using a instrument called circular dichroism instrument, okay, or CD instrument. Okay, and this kind of spectra would be called CD spectra. Okay, so we will see some characterization of biomolecules in CD spectra in a short more in a few more lectures, and you can use to identify the the various conformations that are present in biomolecules, like alpha helix, beta sheet, the various forms of DNA, and so on. We'll see that they are optically, and they have different optical and 3D structure from each other. So what is second aspect is, what is the utility of this chirality and enzymatic chemoselectivity. So this is probably more of a practical use. So this aspect, that this molecule have different arrangement of groups in 3D space, is utilized by biomolecules in chemical enzymatic selectivity. Okay, so let's give an example. So let's just talk about adenosine. Okay, so or DNA synthesis. So in the DNA synthesis, what happens is that the polymerase is the enzyme, the DNA synthesis, which is is part of the process called replication. So we could have taken RNA synthesis also, but let's just talk about DNA synthesis. So let's just take a simple base called adenosine. Okay, so what happens here is the adenosine is part of a growing phosphate sugar chain. Okay, so this part, there are other bases. This is phosphate, and this is your sugar, deoxyribose sugar, and this is the base. So if you recognize or try to look at this, so this particular carbon is a chiral carbon. Okay, has the 4 different substitution: oxygen, hydrogen, carbon, and nitrogen. So in the polymerization reaction or polymerase replication, what happens is, if you have a, what the polymerase does in the opposing chain, it incorporates a T, right? Then if you have a C, it incorporates a G. If you have a T, that incorporates a next to it. Okay, if you have a, then it incorporates another T, like that. Okay, so that is how it does. And the defining factor is the hydrogen bonding. So how it recognizes this two? There is a specific hydrogen bonding between two, specific hydrogen bonding between two. That is how it ensures the chemoselectivity. Okay, so this leads to chemoselectivity, that it does not misincorporate any wrong base. But there is the stereoselectivity aspect also there. So let's say it has to, let's say we are giving the, so we are trying to incorporate T opposite to it. So T will come opposite to it, and the T is going to look like something like this. Okay, so that would be this particular base, NH CH3 carbonyl and base, and what will happen here is, this is your the opposite strand, and this is the growing sugar. Okay, this part of the triphosphate, and if there is a match, it will, so if there is a hydrogen bonding match, it will lead to breakdown of this phosphate and only incorporate until this part in the opposite chain. Okay, so that is how it incorporates that, it makes sure that there is a hydrogen bonding match and the chemoselectivity is ensured. Now imagine you have put the other stereoisomer of the adenosine. Okay, so the other stereoisomer of adenosine would look something like this. So I'm just changing this position, okay, and not the others. So this is how the other stereoisomer is going to look like. So hydrogen would be on the top and the nitrogenous base would be on the bottom side. So now you can imagine that if your base is base is placed on the opposite side, it can no longer match with the T. It can no longer match with the T because T is present in the enzyme in the cavity or the active site like this. So this is presented by the T to the a growing chain like this, and it would lead to the recognition, right? But if it is placed oppositely, then it can no longer recognize and there will be no hydrogen bonding and the chain will not grow. So you need to have a oppositely placed T, which is non-natural T, for this to work. Okay, but this conformation, once it is reversed, this will no longer have a hydrogen bonding with natural DT, DTTP, okay, deoxythymidine triphosphate, okay, because the relative arrangement of the groups is and the 3D space is changed, okay. So this will no longer be able to do it. So if you put the T here, okay, the T will somewhere be here, right? So let's just draw it out. Going to be a bit messy drawing. So the T will somewhere be here. Okay, so the T will be somewhere here, presented by the enzyme, and your base, but now your base is no longer present. Okay, your where you cannot do participate in a hydrogen bonding anymore, right? So your base is, you cannot do any more hydrogen bonding, so no hydrogen bonding and no replication. So because this will not change, because enzymes have a defined 3D arrangement, because all the enzymes, again, their chirality is fixed. The DTTP has a defined 3D arrangement, so because it is also fixed. So just changing this will completely shut down the replication process. I mean, if you have to then redesign the enzyme, redesign DTTP to get this done. Okay, so that is why the enzymatic chemoselectivity or stereo-selectivity is dependent on the relative arrangement of atoms in the space or chirality. Stereo-chemoselectivity. Okay, so that is how it is defined. Okay, so the last thing that I want to talk about is enantiomers. So enantiomers are what are enantiomers? So these are stereoisomers, but here what you have done, you have changed the atoms 3D arrangement for a chiral carbon. Okay, so this is a complicated slide, so I'll just draw one myself. So enantiomers are, let's say you have a chiral molecule here, okay, having A, B, D and E, and you have switched any two atoms, okay, that is B and E is switched. Okay, so this is called an enantiomers. Okay, so this is a chiral carbon, you can recognize now. This is also a chiral carbon, but these are enantiomers. So what is enantiomers? So these are molecules which are mirror images, chiral molecules. Okay, so I should use the term chiral. Molecules which are mirror images of each other. Okay, so this is, so how is this a mirror image? If you put a mirror here, so that would be, you can see that this is completely reflected. Okay. Similarly, if you take the same molecule, let's call this 1 and this call this 2. If you take molecule 2 again and put a mirror below, that would lead to the same enantiomer being formed. Okay, this is chiral carbon and we are putting a mirror here. Okay, this is going to interchange of the D and A, but not E and B. Okay, so this again, we will be able to prove that this is molecule 2 again. Okay, so this is a enantiomers. Okay, so irrespective of where you are putting the mirror, you are going to get same enantiomer. So each chiral molecule has only one set of its enantiomers. Okay, so these molecules, what is the specialty? These are non-super-imposable, meaning you can see if you try to bring and put this on this molecule, the E and B will not be placed next to each other, but A and D will. Okay, so what I will do, I'll take a blue color and draw this molecule on top of this. So you can see here, if I draw the A and D, and if I'm going to draw the other part, that is E and B, the B and E are not matching. A and D, they are superimposing on each other, but not the B and E. Okay, so these are non-superimposable mirror image of each other. Non-superimposable mirror image of each other. So we'll prove something in a moment, that this aspect, this non-superimposable mirror image, is only true for if it's an enantiomers or if it's a chiral molecule. Okay, the second point is they have identical chemical properties. The third point is they have different optical properties, meaning if one reflects the plane polarized light in this direction by 60 degree, the two will reflect it in the opposite direction by 60 degree. Okay, so if it would be a minus, okay, because it's in the opposite direction. Okay, so reacts or interacts differently with plane polarized light of that, and leads to opposite angles and directions of rotation of the plane polarized light. Okay, so we just will prove something in the using 3D image, that this non-superimposable mirror image, it will not be true if it is not a chiral molecule. Okay, so we'll just quickly show a new slide and quickly prove it. So first I will show you using just a drawing, then we'll go to the 3D model. So this molecule, if it's a B and D, okay, this is not chiral, right? Because they do not have identical chemical, non-identical 4 valences, right? So if you do a reflection of it in the mirror, this will lead to A and B in its own place, and A and D will be interchanged, right? So this is not enantiomers, and the stereochemical term means these are homomers, meaning these are identical molecules. Okay, and second point, these are superimposable mirror image, meaning because these are identical molecule, you can place them on top of each other. Superimposable mirror images. Okay, so what we will prove now using Avogadro model, that if you have a chiral molecule versus not chiral or achiral molecule, so this leads to this its mirror image leads to non-superimposable mirror image superimposable mirror image or enantiomers, as opposed to here its mirror image leads to superimposable mirror image. Okay, so that will do the first thing in the next lecture. Okay, so that will prove it. Okay, so I'll stop this lecture now and we'll prove that in the